friends, now I will start with another hormone, which is abscisic acid. Now till now we have discussed about auxins, gibberellins and cytokinins, which are considered as the growth hormones. Abscisic acid is a class of hormone which is considered as an inhibitory hormone because it prevents or inhibits the growth of the plant. Now this hormone is also known as a stress hormone because it allows the plant to pass on or to cross through the stressful conditions or to pass on through the stressful conditions, whether these stresses can be biotic or abiotic. So such kind of hormone will give the plant strength to pass on from these stressful conditions. So this hormone basically makes the plant to undergo dormancy, thereby by which the plants can cross through the stressful condition, and when the favorable conditions are back, again the production of this kind of hormone decreases and the growth of the plant is retained. So it all began in 1961 when Louis and Carins they isolated a crystalline substance which they named as abscissin or later abscissin 1 from the cotton burrs, because there was a problem in their area that cotton burrs were keep on falling from the plant. So this make them curious that what is the reason why these cotton burrs are falling from the plant. So they start working on the cotton plant, and while working they isolated this compound, and since this compound was responsible for the abscissin of the cotton burrs, they gave the name abscissin. Though they identified the properties, that is, this compound was responsible for the falling of the cotton burrs, but they were not able to elucidate the structure of this compound. Now their discovery was followed by the discovery of Okuma et al. In 1963 when they isolated a similar type of compound again from the cotton burrs, but they gave the name to this compound as abscissin 2. And they also gave the tentative empirical formula, that is C15H20O4. Now since they gave this empirical formula, this gave an idea to the scientific community that this compound could be a terpenoidal compound in nature. Though they were not having the exact structure, but they isolated the compound. They were not even knowing whether the abscissin 2 or abscissin 1 compound are same or not, but both these compounds were having similar kind of property and they were responsible for the falling of the cotton burrs from the cotton plant. In the same year, Eagles and Warren they again isolated another compound from the downy burrs, that is Betula pubescens, and they named this compound as dormin because they found that this compound is responsible for the inhibition of the growth of the plant. This type of the compound is responsible for the induction of dormancy in the plants, therefore they gave the name dormin. And even they were also not able to elucidate the structure of this compound dormin, therefore they sent this compound to the Shell Research Laboratory in England for its spectral analysis. And in the meantime they also compared the activity of dormin with the abscissin 2 compound in 1965, and they found that dormin as well as abscissin 2, both these compounds were having similar kind of activity when in terms of abscission of the plant parts. And in 1965 Okuma he isolated the structure of abscissin 2. Now lot of confusion started to develop in the scientific community because all the three compounds, abscissin 1, abscissin 2 and dormin, they exhibited similar kind of property, similar kind of activity. So whether to give the name abscissin 1 or abscissin 2 or dormin, so what should be the name given? So a lot of confusion developed. Therefore in 1961 all the groups who were working on these compounds, Edicott's group, Waring's group and the Shell groups, they came together in a international conference on the plant growth regulators, and they unanimously decided to give a common term to this compound, and they found that all the three compounds, dormin, abscissin 2 or abscissin 1, are nothing but they are a similar compound, so they unanimously agreed to give the term abscisic acid for all these compounds. And this is how basically the term abscisic acid came into existence. Now what is abscisic acid? As I told you that it is a 15 carbon compound so which is a weakly acidic in nature and is sesquiterpenoidal in nature. Now these compounds are related to monoterpenoids or diterpenes or triterpenes or even carotenoids by virtue of their biosynthetic pathway, because these compounds along with the terpenes, they share almost similar kind of biosynthetic pathway, which we are going to discuss ahead. Now these compounds, as I told you, they exhibit or regulate various physiological and biochemical processes in the plant, and they are known as inhibitory hormone because they allow the plant, they inhibit certain activities in the plant which allows the plant to pass on to the unfavorable condition which can cause biotic or the abiotic stresses on the plant. So these are also known as stress hormone, as we have discussed it earlier. Now there are certain essential structural features for these abscisic acids. First one is the free carboxyl group, which is present in the structure. This is essential for their activity. Apart from the free carboxyl group, they should contain a cyclohexane ring, and not only cyclohexane ring, but they should have an unsaturated cyclohexane ring, and this unsaturation or the double bond should be present between the second and the third carbon in the cyclohexane ring. And not only the double bond, but this double bond should be present in the cis configuration, because the trans configuration will eventually lose the activity. And these abscisic acid molecules or the compound are stored in the plant in the form of glycosides. Whenever the glucose is attached to the abscisic acid molecule, and this glucose molecule is attached at the hydroxyl group which is present in the acidic moiety of the structure, so the abscisic acid, when it is present in the glycosidic form, glucose molecule attached, this form act, or the glycosylated form act as the storage form of the abscisic acid. And whenever this glycosylated form undergo hydrolysis, thereby removing the sugar molecule or the glucose molecule from the carboxyl group, releasing the free abscisic acid, which is now active to show its physiological effects on the plant. Next we have the biosynthesis of the abscisic acid. Now earlier it was, since the abscisic acid is related to the terpenoidal compounds, earlier it was believed that it is synthesized from the mevalonate pathway, which was considered as the direct pathway of the synthesis of abscisic acid. But later it was found that in plants specifically, abscisic acid is synthesized through the methyl erythritol phosphate pathway, that is MEP pathway, via isopentenyl pyrophosphate and carotenoids. So isopentenyl pyrophosphate is formed and carotenoids are formed before the synthesis of abscisic acid, and therefore it is also known as indirect pathway. Whereas synthesis of abscisic acid through mevalonic acid pathway or mevalonate pathway was found in fungi. If we look into the overall biosynthesis of abscisic acid, we will find that the biosynthesis of abscisic acid is a two compartment synthesis or the two compartment system, as it partially occurs in plastid and partially it occurs in the cytoplasm. So if we talk about the steps that occur in plastids, first step is the isopentenyl pyrophosphate, which is a 5 carbon moiety. The 3 moieties of isopentenyl pyrophosphate, they join together to form or to give rise to a 15 carbon moiety, that is farnesyl diphosphate. And to this 15 carbon moiety, another isopentenyl pyrophosphate molecule joins to give rise to a 20 carbon moiety, that is geranyl diphosphate. Now the two geranyl diphosphate molecules come together, they join together and give rise to a 40 carbon moieties, which are known as carotenoids, and beta carotene is one of the most common example or the most common carotenoid that occur in the plants. Now this beta carotene then gets converted into zeaxanthin, which is again a 40 carbon moiety, and this zeaxanthin, which is acted upon by zeaxanthin epoxide and gets converted into a trans violaxanthin via antheraxanthin. So this zeaxanthin is acted upon by ZEP enzyme, which converts first and into an intermediate which is known as antheraxanthin, and finally into trans violaxanthin. Now this trans violaxanthin then gets converted into cis-neoxanthin, or it can first get converted into trans-neoxanthin and finally into cis-neoxanthin. Then this cis-neoxanthin is oxidatively cleaved by 9-cis-epoxy-carotenoid deoxygenase and it produces xanthotoxin, which is a 15 carbon moiety. So we started with a 5 carbon moiety, we reached to 40 carbon moiety, that is carotenoids, and eventually these carotenoids that then get degraded by various enzymatic action, and we are back to a 15 carbon moiety, that is xanthotoxin. And this step of conversion of cis-neoxanthin into xanthotoxin is considered as the major rate limiting step in the biosynthesis of abscisic acid. This xanthoxin is then moved outside the plastids into the cytoplasm, that is the second component of the biosynthesis of abscisic acid. Xanthoxin in the cytoplasm is acted upon by alcohol dehydrogenase and gets converted into abscisic aldehyde, which is eventually converted into abscisic acid by the action of abscisic aldehyde oxidase enzyme. So this is how the abscisic acid is produced in the plants. So this is nothing but a diagrammatic representation of the biosynthesis of abscisic acid. You can see that it starts with the isopentenyl phosphate, that is 5 carbon moiety, to 15 carbon moiety, to 20 carbon moiety, that is geranyl diphosphate. Two moieties get combined to give rise to carotenoid. Carotenoid gets converted into viola-xanthin gets converted into neoxanthin to xanthoxin. Xanthoxin moves out of the plastids into cystol, and it converts into abscisic aldehyde, which finally gets converted into abscisic acid. Now this abscisic acid is stored in the vacuoles in the glycosylated form. So glucose gets attached to the abscisic acid molecule, and this glycosylated form is eventually stored in the vacuole, and whenever there is a requirement of abscisic acid or unfavorable conditions are there or any stress is there on the plant, this glycosylated form of abscisic acid is released from the vacuole, it gets hydrolyzed, and abscisic acid exhibit its effect. Now we move on to the signaling of abscisic acid or the signaling pathway of abscisic acid. So as I told you that abscisic acid is generally a stress hormone, and it provides a robust response to the environmental stress such as high salinity of the soil or extreme temperature condition, whether it is chilling or it's very hot, and if there is a water scarcity, drought is there, or a dehydration of plant cells occur. So such kind of stresses causes the increase in biosynthesis of abscisic acid, or they causes the hydrolysis of the glycosylated form of abscisic acid, and that abscisic acid is released from the vacuole. And once the abscisic acid is released, it exhibits its inhibitory effects, thereby allowing the plant to cope up with these kind of environmental stresses. Now if we look into the signaling mechanism of abscisic acid, there are various components that are involved in this signaling pathway. And the first and the foremost component is the abscisic acid receptors. Now there are different types of receptors for abscisic acid that have been identified, like pyrabactin resistant, that is PYR type of receptors, PYR or pyrabactin resistant like receptors, that is PYL receptors, and there are 13 subtypes of PYL receptors have been found. Third class is of regulatory component of abscisic acid receptors or RCAR receptors, and 14 subtypes of RCAR receptors have been identified. Another type of receptor is the chloroplast protein or CHLH receptors. Earlier these chloroplast protein receptors were found responsible, or it was believed that these receptors are responsible for the chlorophyll production in the plants. But later it was found that these receptors have specific capability of binding with abscisic acid, so therefore now they are considered as abscisic acid receptors. And last one is the G protein coupled receptors or the GPCR type of receptors, and specifically ATGT-G1 and ATGT-G2 type of receptors that have been found in the plants. Amongst all these receptors, PYR, PYL and RCAR are the three main type of abscisic acid receptors which have been worked upon extensively. The second component is the surface proteins, which are specifically the protein phosphatase 2C or PP2C components. Third component is the SNF1 related protein kinase 2, that is SNRK2. Fourth component is ABRE binding proteins, that is AREBs or abscisic acid responsive element binding factors, that is ABF. And eventually the last component we have is the ABA responsive elements, that is ABR. SNRE. Now, protein phosphate 2C, that is PP2C, and SNRK2 proteins, or SNRK or the protein kinase 2, they play an important role in the induction of the abscisic acid, because as we will discuss further, these protein phosphatase and SNRK, they are bound together before abscisic acid acts on the abscisic acid receptors. So here you can see how that abscisic acid exhibit its effect. Under the non-stress condition or under the absence of abscisic acid, the receptors are not bound to abscisic acid, the receptors are free. And when receptors are free, the protein phosphate, that is PP2C, it binds to SNRK2, that is protein kinase 2. And when SNRK2 is bound to PP2C, it is not able to exhibit, or it is not able to activate or inactivate or inhibit various kind of response elements. And when the SNRK is not able to activate or inactivate various response elements, no effects are exhibited in the plants. But just now imagine the presence of abscisic acid, or under the stress condition when abscisic acid is present. Now when abscisic acid is there, obviously it will bind to the abscisic acid receptors. Now as soon as the abscisic acid binds to its receptor, there occur certain conformational changes in this complex, and by virtue of these conformational changes, now this complex become more prone to the binding of PP2C proteins. Or in other words, we can say that abscisic acid and its receptor complex has more affinity for PP2C proteins in comparison to SNRK. So what will happen now? This phosphate proteins or PP2C proteins now will bind to the abscisic acid receptor complex. So now SNRK2 is free for phosphorylation. It will undergo autophosphorylation, or various kinases causes the phosphorylation of SNRK2 proteins. So once these SNRK2 proteins are phosphorylated, they activate or deactivate certain response elements, such as they will inhibit Ka-T1 channels, which are nothing but the potassium influx channels. These channels are inhibited. They will activate the SLAC1 ion channels, which are slow anion channels. They will also activate the gene transcription factors, which we have discussed, that is ARV or ABF. They will also stimulate or activate the transcription factor AB15. And apart from that, they will also activate certain enzyme which will exhibit the effect on the metabolic pathways of the plant. So all such response elements, whether they are activated or they are inhibited, they collectively exhibit various effects. They collectively upregulate various genes that will exhibit the effect of abscisic acid. Now we move on to the actions that are exhibited by abscisic acid. So as I told you that this is a stress hormone, so it controls the transpiration rate of the plants under stress, specifically under drought condition, by closing the stomata. Because transpiration, apart from gaseous exchange, stomata are also responsible for the transpiration of the water. So under drought condition, water scarcity is there, so stomata close, thereby controlling the transpiration and preventing the dehydration of the plant. Abscisic acid also induces the bud and seed dormancy. So it causes the seed maturation. It also prevents the germination of the seed under unfavorable conditions. It stops the protein and RNA synthesis in the leaves, thereby causing the leaf senescence. So as soon as the unfavorable condition occurs, we see that leaves start falling, and this is largely due to the excess of abscisic acid that is produced inside the plant. It also stops the cambium activity under harsh condition, thereby allowing the plant to enter into the dormant phase or under the dormancy. It also causes the abscission of the flowers and fruits, because this was the basis for the initiation of the discovery of abscisic acid, wherein we discussed that the falling or the abscission of the cotton burrs that lead to the discovery of these important plant hormones. So I already told you that they also lead to the seed maturation. Now they have an antagonistic action against auxins and cytokinins. Obviously auxins and cytokinins are considered as growth hormones, whereas abscisic acid is considered as an inhibitory hormone, and therefore we can say that abscisic acid activity, it will help in keeping the activity of auxins and cytokinins under check. Now I will discuss the mechanism of some of the important physiological effects that are exerted by the abscisic acid, and the most important one is the stomatal closure. So as you can see in this diagram, there are two different pathways that have been proposed by which abscisic acid leads to the closure of the stomata. One is the calcium dependent and other one is the calcium independent pathway. Now under stress conditions or under drought condition, when there is scarcity of water, abscisic acid production increases in the plant. Now this increased production of abscisic acid will lead to the production of certain reactive oxygen species, and these reactive oxygen species or ROS, they will activate or they will open the calcium influx channel, thereby allowing calcium ions to enter into the guard cells. Now once these calcium ions enter into the guard cells, they exhibit various other effects. Now before I move on to those effects, the abscisic acid whose concentration increases inside the guard cell, it also activates isopentenyl triphosphate. Now this isopentenyl triphosphate, it causes the activation of calcium channels that are present in the vacuoles in the guard cells. And when these channels get activated, the calcium ions that are present in the vacuole, they start moving outside the vacuole, and the concentration of the calcium ions increases in the guard cell by virtue of these channels in the vacuole and by virtue of the influx calcium channels that are activated by ROS. So once the concentration of calcium ion increases, they inhibit the proton pump and thereby inhibiting the movement of proton ions outside the guard cells. So we can say that there is a substantial increase in the positive charge inside the guard cell, because calcium ions are also positive charge and proton ions are also positive charged. Now this increased calcium ion concentration further activates certain calcium dependent protein kinases, and these calcium protein kinases, they activate the slack anion channels, and these channels once get activated, they cause efflux of the chloride ions, whereas they also activate certain coax channels which causes the inward movement of the potassium channels, and they inhibit the Cat1 ion channels, which leads to the efflux of the potassium channels. So all in all, we can say by increasing the concentration of calcium ions in the guard cell, there is disruption of the osmolarity of the guard cells, and to balance this disruption of osmolarity, water start moving outside the guard cells, and as soon as the water start moving outside the guard cell loses their turgidity, which eventually leads to the closure of the stomatal pore, and this is how it prevents the dehydration or the loss of water through transpiration. In the calcium independent pathway, OST, that is open stomata 1 channels, are activated or they get phosphorylated, and these cause activation of certain QAC1 ion channels, which causes the efflux of malate ions. They also produce ROS, which causes the efflux of chloride anions through SLAC1. They also inhibit Cat1, which prevents the influx of the potassium ions. They also prevent certain other anionic channels, which again causes the disruption of the osmolarity and eventually causes the movement of water outside the guard cell, causing the loss of turgidity and closure of the stomata. Amongst these two calcium dependent pathways, most extensively studied and is considered to be the major reason behind the closure of the stomata. Now the another major effect is the seed maturation and dormancy, and the three major steps that are exhibited by the abscisic acid is the prevention of germination, degreening of the seeds, and the desiccation tolerant, that is the accumulation of the food in the seeds that will allow the seed to undergo the dormant condition, and whenever the favorable conditions arise, this food is utilized for the germination. So as you can see, as soon as the abscisic acid binds to the receptor, it makes SNRK2 proteins free to phosphorylate, and when the SNRK2 protein phosphorylate, they cause activation of transcriptional factor AB5. This AB5 further activate transcriptional factor AB3, and this AB3 prevents the germination of the seeds. Now AB15 and ABI3, they further activate ABI4 transcriptional factor, which causes inhibition of CYP70A1 and A2 enzyme, which are responsible for the germination of the seed. And when these enzymes are inhibited, germination also gets inhibited. So this is how abscisic acid prevents the germination of the seeds. Now AB15 and AB13 also inhibit certain genes which are known as stay green genes, such as SGR1 and SGR2. And inhibition of these or down regulation of these genes causes the degreening of the seed, because before the seeds get mature, the green color is lost by the seeds and they get converted into a brown colored seed, which allows them for their storage. So this is how they causes the degreening of the seeds. And last is the accumulation of the food. So as I told you that abscisic acid causes the activation of transcription factor AB15 and AB13. They activate the dog proteins, that is delay of germination proteins, and these proteins further activate LEA proteins, that is late embryogenesis abundant proteins. These proteins causes the accumulation of nitrogenous rich food material within the endosperm of the seed and allows the seed to go into dormancy, and whenever favorable conditions arise, this food material that is stored in the seed endosperm is utilized by the seed. The CYP70A1A2 enzymes, they get activated because now abscisic acid is absent, and that eventually leads to the germination of the seed. So this is, friends, this was all about the abscisic acid, how it is biosynthesized in the plant cell, what is the signaling pathway of abscisic acid, and how it exhibits its various physiological effects in the plant cells. Thank you.